In lab activity number 1, we looked at various aspects of behavior of light, the light coming from a laser, as well as image formation. So let's go through the different aspects of this particular activity and discuss what they illustrate. In the first one, you had a green laser. Your first placement of the laser was on the side. So you aligned the laser with the side of the beaker. On the opposite side of the beaker, you could put your hand or you could put a card acting like a screen. In this situation, when you turn the laser on, a green beam emerges from the laser. It then travels through the water, out the other side of the beaker, and it hits your hand or your card, and a green dot appears there. So the dot appeared straight across from where the laser was positioned. And of course, this happens because the laser light is entering along the normal. The angle of incidence is 0 degrees, so there is no apparent refraction. In the second situation, you took the laser and you aimed it from the side, pointing it up towards the ceiling in this fashion. So essentially, we're aiming it towards the underside of the water that's in the beaker. Again, you turn on the laser, the green beam emerges, and it travels to the side of the beaker. We can draw normal in, so we see the angle of incidence. We know that when light enters a more dense medium, that it refracts. In this situation, it refracts towards the normal, as you see here. It is now traveling towards the undersurface of the water, and it hits at a fairly large angle when you look at the angle of incidence to the surface. This angle is so large that, in fact, it simply bounces off the underside and totally internally reflects. So it continues towards the side, the far side in this case, of the beaker. The angle this time is shallower, so when it hits the side, it is actually able to emerge, and going to a less dense medium, it bends away from the normal and continues along that path. You would have picked this up on your hand, or if you put a card there, the beam on that side would have been able to emerge from the water. In the third positioning of the laser, you placed it from on top, aiming it downward towards the top surface of the water in the beaker. You turn the laser on, the green beam emerges, and it continues till it hits the surface of the water. You're going from less dense to more dense medium, so you expect it to refract towards the normal, which is exactly what it does. It continues on that pathway till it gets to the side of the beaker. Now we're trying to go from more dense to less dense medium. It's a fairly sharp angle, so we've exceeded the critical angle, and the light beam simply bounces off the interior wall of the beaker. I then asked you to place a dry finger against where the dot is appearing, and when you do that, it has no impact on the reflected beam, the internally reflected beam, and things stay as they were originally. Now, what if we look at that situation again? So again, I have the beam aimed towards the top surface. It hits, it refracts towards the normal, It continues on its pathway to the side. The angle is too large for it to emerge. It bounces off the interior surface. But this time, if you take your finger and you wet it first, when you press it against the dot at the side of the beaker, voila, the beam does not continue to internally reflect. It actually emerges because you've altered the density, and you should have seen your finger completely illuminated by the green beam. We took away one of the key conditions for total internal reflection, and that, of course, is the density. It only happens if you go from more dense to less dense. The little bit of water continued the density of the water in the glass, and so it no longer totally internally reflected. Finally, you dealt with a penny that was placed inside of a beaker of water. You're looking down upon it, and you saw three images. Let's analyze those three images. The first image is close to where the real penny really is. So some of the light coming off of the penny is going to be trying to continue through the water and come out of the beaker. So it travels to the side of the beaker. We're going to go from more dense to less dense. So it bends away from, it refracts away from the normal. And some of those light rays, therefore, will continue on and enter your eye, and you'll form an image of the penny. Now, your brain is going to trace that ray from where it thinks it came from, which is here. And so at that position, it will form an image of the penny. So you can see that first image is actually a little bit closer to the front of the beaker and is in fact not where the penny really is located. Now, on the other hand, there are other light rays emerging from the penny heading away from you, and they're shooting towards the side of the beaker, and they're coming in at a fairly large angle. And so the angle of incidence is too large for it to escape from that side, and it totally internally reflects, heading up towards the surface of the water that's in the beaker. Now, this time the angle is shallower, so the light is able to escape. It refracts away from the normal, continues on its path to your eye. Your brain, again, does not trace it off of all those anglings that took place. It thinks it came from a straight line position, and this time your brain forms an image on the side of the beaker. So that is the second image of the penny. And finally, there are a set of light rays headed towards the surface of the water in the beaker, strikes that undersurface. The angle is pretty shallow, so it is able to escape. It bends away from the normal as it went from more dense to less dense. It continues on to your eye and hits your retina. Your brain traces that ray from where it appears to have originated, which is a position up towards the top surface of the beaker of water. And there it is. So that's how you end up with three images of the penny. Now you had to position yourself until you saw three of them. You don't see three images at every single position because in some situations you're just not going to have the proper angle to allow those formations to occur. Now, another area that you looked at in this activity was the pencil. So you took your pencil and you placed it in a beaker. So here's your beaker of water. And in the first instance, you simply placed it right in the very center of the water. And you noticed that the bottom half of the pencil was magnified. It aligned with the top of the pencil, but it was larger than the top half, so it seems thicker on the bottom. If we look at that from a top view so we could trace light rays. There's your pencil in the very center of the beaker of water. Light rays are going to therefore emerge along any of the normals, depending on where you're positioned. There's the normal. Here you are getting ready to view any light rays that come off of the pencil. So it travels right along the normal. The angle of incidence was 0, and it hits your eye. And because of the shape of the beaker, the water is forced into this convex shape, which is typical for lenses that magnify images. And that's why you see a magnified image. We will explore this in detail at a later time. Now, you then took that pencil, here again is your beaker of water, and you moved it across your field of vision to the left and to the right. Here you see it's been slid over to the left. And you notice now what's happening is the bottom again is still magnified because of the shape of the beaker, but the bottom is no longer aligned with the top half of the pencil. It seems to be disjointed and it seems to be off to the side. So let's view that from on top and let's see if we can trace the rays and figure out why this is happening. So here you see the pencil off to the side. It's no longer in the center of the beaker. Light rays are going to come off of the top half and the bottom half, but we're focused on the bottom half of the pencil because that's where this effect is happening. There's my normal. The ray travels and it hits the side of the beaker. It's about to go into a less dense medium, so it refracts away from the normal. You're positioned to capture some of those light rays. They continue as they emerge from the beaker to your eye, hits your eye. Again, your brain processes that information and it thinks that light ray originated over here. And so the image of the bottom half of the pencil appears to be to the left side, to the outer side of the beaker, and so therefore does not align with the top half of the pencil where the light rays never changed media and therefore never refracted. So that's why you see a disjointed pencil. It's because you're no longer coming along the normal, you're coming at an angle. Now, total internal reflection is something that you can observe in everyday life. And here is a very cool picture where you see a swimmer in crystal clear water moving off the side of the swimming pool with this big, large flipper. And if you look towards the top of the photograph, you can actually see her reflection on the underside of the water that's in the pool. And so the bottom surface of the pool water is acting like a mirror. The light, obviously coming off of her back and her head, is totally internally reflecting, and you're in the right position, just like you would for any other mirror, to see that reflected image, and it reaches the lens of the camera, and we're able to capture the image. That was lab activity 1, and hopefully you got a better appreciation of how your retina and your brain work together to form images of objects.